Okay. Emily, can you, okay, so we know Emily's there. Okay, so we're going to start with Emily, right? Okay. Are we on? Murray?
Yes, we are recording. We are good to go.
All right, thank you. All right. Well, welcome everyone to the Undergraduate Research Symposium. This is Thursday, May 27th, and this is It's a Bio Thing. These are presentations by students who are going to be talking with us about their biological research. So we really look forward to that today. I'm Barbara Jenkins. I'm a research librarian at the University of Oregon, and I am going to be the initial moderator, and I'm co-hosting with Marie, and she's going to be taking care of all the Zoom technical issues. So panelists or anyone else that has any problems, just to put something in the Q&A or signal to Marie by raising your hand that you're having any problems, okay? All right, well, we're going to follow a format which allows us to have a presentation followed by a few questions, and then we will have plenty of time at the end for questions, additional questions as well. But we have 4 panelists, and so we have plenty of time for discussion right after the person's presentation. So we're going to start today with Emily Cook, and she's in environmental science, and she's going to be talking about the intraspecific variation in a particular form of bluegrass resistance to annual grass invasion. Emily, it's all yours.
Awesome, thank you. Let me share my screen.
Emily, I just wanted to let you know that you have 10 to 15 minutes, as you know, and at 13 minutes, I will just raise my hand just to let you know that you're 2 minutes from your time, okay?
Awesome, thank you. Okay, hello everyone, my name is Emily. I'm really excited that you all are joining us for this webinar to share our thesis results and other awesome experiments. I'm really excited to share the research that I've been working on for the last few months on plasticity of traits in a native grass in competition with exotic species. So I will just jump into it. Exotic species are a serious threat to ecosystems across the Western United States. Exotic species invasion is second only to habitat loss and degradation as leading cause of biodiversity reduction. The species of interest to me and to many rangeland managers and ecologists in the Great Basin is cheatgrass. Cheatgrass invasion in combination with a change in fire regime and climate exacerbate large fires in the region, which in turn results in loss of native plant species, including the native perennials, which are excellent competitors of cheatgrass invasion. A prominent question for restoration is how to choose competitive native plants and which traits those plants should show in order to compete with invasion for successful restoration. In this study, I explore how within species trait variation may create an advantage for native grasses in competition with cheatgrass in a region where shifting climate is also altering water availability. So to give an outline, I'll briefly provide some background knowledge to provide context for my study and go over the methods, my analysis thus far, and the initial conditions. So one major obstacle to preventing successful restoration in regions where cheatgrass is prevalent is a shift in fire regime. Cheatgrass has quickly altered the fire regime across North America, increasing the fire size and frequency. Previously, low intensity fires were utilized by the Paiute people to reduce fire fuel, but Euro-American settlement has largely suppressed fire, leading to catastrophic megafires. This change in fire management paired with shifting climate conditions make the Great Basin grasslands even more susceptible to cheatgrass invasion. A shifting climate may also favor cheatgrass establishment in the future. Climate is highly variable across time and space in the Great Basin, and spring and summers in the future are predicted to be hotter and drier, as well as an extended fire season, while winters are predicted to be increased in precipitation. This is important because cheatgrass is a winter annual whose germination occurs in early fall and winter due to increased precipitation, and an increase in temperature and altered precipitation will likely be favorable conditions for cheatgrass. So to prevent more of these huge fires and invasion of cheatgrass, seeding of native perennial grass species has been adopted as a management strategy for post-fire rehabilitation. The promising candidate that I've been looking at is Sandburg bluegrass. This is a native grass species known to have high variability in traits across climate and space. So some populations of POA may be better at resisting cheatgrass than others. Seedling traits in particular may affect their ability to resist invasion by cheatgrass. So in this study, I hypothesized that POA's resistance to cheatgrass will decline with reduced water availability, but POA populations from warm and arid source environments will have smaller decline than those from cooler and wetter environments. I also hypothesized that POA will show variation in traits across populations, and I hypothesized that POA populations with greater trait plasticity will better resist invasion to cheatgrass. This is a map of the site locations where seeds were collected in the Great Basin. The boundaries of the Great Basin are in black and the site locations are in orange. My study was a greenhouse experiment on campus at the University of Oregon, where we had cheatgrass in competition with these 6 populations, which were from several sites in Oregon, California, and Nevada. Each population of POA was sown in monocultures and in competition with cheatgrass. And additionally, we had a wet and dry treatment where wet trays were watered more than dry trays. Due to the wide array of traits that a plant may display, the question of which traits to measure becomes important. However, plant size, seed size, and leaf structure are commonly considered pertinent due to their significant influence on plant performance and ease of measurement. So the data that we collected on traits for POA were stem count, above ground biomass, specific leaf area, leaf dry matter content, height, and seed mass. So to investigate my first hypothesis that POA resistance to cheatgrass will decline with reduced water availability, we used emergence and biomass as a proxy, where fewer stems emerged and lower biomass will relate to decreased resistance to invasion. In this first figure, we first established that POA showed germination in both presence and absence of cheatgrass. When in competition with cheatgrass, there's no difference in emergence between wet and dry treatments except for in Reno, where emergence was greater in wet than dry. This second figure shows that in both competition and non-competition pots, wet treatments have a higher mean dry biomass weight than dry treatments, excluding gund. And there's no difference in mean dry biomass weight across populations within both dry and wet treatments. So to address my first hypothesis that resistance would decline with decreased water availability, when we look at emergence, we can't really say that this is true. We can say that water availability has an effect on emergence of POA when it's not in competition with cheatgrass. But when it is in competition with cheatgrass, it's essentially the same as saying it's in a drought year. So when looking at biomass, there is a likelihood that resistance to cheatgrass declines with reduced water availability, since the wet treatments have a higher dry biomass than the dry treatments. And additionally, the wet treatments without competition is higher than the wet treatments with competition. However, POA populations from arid source environments didn't necessarily have a smaller decline than those from cooler and wetter environments. To address my second hypothesis that POA would have variation across traits, we looked at the means and standard errors of leaf height, dry biomass weight, leaf dry matter content, specific leaf area, and seed weight. And the main takeaway here is that there is variation across populations for each trait. You can see that some populations have pretty similar variation, but for the most part, there's pretty clear variation in traits across. For example, in the dry biomass weight, you can see Butte Valley showed the greatest mean dry biomass weight, while Gund had the lowest. Okay, so to begin to address my final hypothesis, I use Pearson's coefficient of variation to look at the plasticity for each trait by population to see which traits in which populations have high variability. So dry biomass weight showed the greatest amount of variation and is the most plastic trait measured. And the other 4 traits show pretty similar variation overall. There's not really a consistent pattern showing that a population might have high variation across traits thus far. But by trait, Steens had a high CV for dry biomass weight. Water Canyon had a high CV for height. Reno had a high CV for leaf dry matter content. And EOARC had high CV for specific leaf area. So the final portion of my analysis, which is not yet complete, will be to determine if populations have high variability, or those which have high variability also have greater resistance to invasion by cheatgrass. And although my research isn't entirely finished yet, I can still speak to some important conclusions found so far. So considering intraspecific trait variation will be important in the future to aid restoration efforts which require seeds for a variety of environmental conditions. This study can also help understand how specific traits in native grasses may be more appropriate than others in specific climates and predicted climates in the future. And finally, if these ideas can be applied in restoration, an environment may be established, which is at less risk for intense megafires, which are predicted to be larger and more frequent in the future. So thank you all so much for listening to my thesis. I just want to thank the Hallett Lab, specifically Lena and Lauren, who both helped me so much, and Undergraves and UROP for funding my research. Thank you.
Thank you, Emily. So it is very interesting regarding this because the West continues to dry out and be susceptible to major fires. So are there any questions from the other panelists or in the Q&A, Marie?
There are no questions currently in the Q&A.
Okay, I have one. Emily, this it sounded, as I understand it, that you use a greenhouse kind of process. Would this be any different, do you think, if you were actually in the field and doing field research versus a greenhouse where I understanding that you try to have the same kind of species and the same kind of environment, but do you think that that makes a difference, greenhouse versus direct field research?
Yeah, I do. I think specifically for this project, when we're looking at trait variation, traits are totally known to be influenced by the environment and by genetics. And so that's things like soil and weather and all those kinds of things would be huge impactors on this project. So we definitely had a very controlled environment where we were controlling the water and like the sun exposure, how much light we were taking in during the day. And so I do think that that's a really important consideration to think about how it would change if we had been doing it in the field.
Mm-hmm. And was this a pandemic related kind of thing, or had you already planned to do it in a greenhouse situation?
The plan was to do it in a greenhouse situation, yeah.
Okay, so you didn't have to adjust necessarily for the pandemic situation.
Right, yeah, exactly.
Great, that probably came in very handy. Does anyone else have a further question?
I have two. Oh, there's also one in the Q&A.
Okay, go ahead, Lukas.
Okay, my first one is a little more fun. I wondered if you got to go out there and collect all your samples or if they were already in the lab. That would be interesting. And then I noticed from when you showed like the plastic traits, Gund seemed to be the most just like I don't know. I don't really know what the stats were on all of them, but it seemed to vary the most between all of the genotypes or like the locations. And I'm wondering what your prediction was, because I also don't remember where Gund was located regionally, on why that might be.
Yeah, great question. I did not get to personally collect the seeds, but I did get to go to the field to see in a location near Burns. So one of the sites in Eastern Oregon, which was super fun. And then awesome eye picking up on Gund. That was one population that I have been considering excluding from the research because the seeds were collected a little bit too early. So they were a little bit, what we think were a little immature. And so we had a lot less seeds, and the seeds were a lot smaller. So our hypothesis there is that they were a little bit immature and just didn't do as well. And so it's possible that maybe they do have a lot more variation, but probably not.
Okay, thanks. Can you read out the one that's in the Q&A, Marie?
Sure. How do you think that climate change will impact restoration efforts and cheatgrass's spread in the future?
Good question, Anna. Thank you. Oh, the question disappeared, but essentially, how will climate change affect it? Climate change is making summers super hot, but it's also making precipitation really variable. And so it's predicted that with increased precipitation in the winter, cheatgrass is going to do really well, especially since they germinate earlier than a lot of the native perennials. And so they'll have that head start in germination and get going before the natives come in when there's more precipitation like that. And then in the summers when it's super hot, it's going to be triggering even more wildfires to happen. So it's kind of this feedback loop where we get more and more cheatgrass invasion and hotter summers, which extend the fire season. So.
Okay, yeah. So you were going to be doing some more research on this. Is that going to be in the summer, fall?
No, I'm actually almost done. I'll be finished in the next 2 weeks. Yeah.
Okay, so you're wrapping up shortly. Okay.
Yes. Yeah.
Thank you very much, Emily. Appreciate it. And our next panelist is going to be Audrey Landis, who's in human physiology. And her talk is about what killed red protein genes in white-blooded Antarctic fish, icefish. So Audrey, it's all yours.
All right. Hi, everyone. Thanks for coming. My name is Audrey Landis, and today I am going to be presenting on what killed the red proteins in white-blooded Antarctic icefish. And this study is about the evolution of globin genes in notothenioid fish and in Antarctic icefish. So notothenioid fish inhabit the southern oceans surrounding Antarctica, and Antarctica is clearly freezing cold with freezing cold temperatures year-round, and since the ocean is so cold, the water remains highly oxygenated, and this creates an oxidative stress-inducing environment. But notothenioids have been able to adapt to live in these conditions, as well as icefish, which are a family within the suborder notothenioids. And what's really unique about icefish is that they don't have hemoglobin. So hemoglobin is a super important oxygen binding protein, and myoglobin is a muscle oxygen binding protein, and they're what make blood and muscles appear red. So this here is an Antarctic notothenioid, and as you can see, its blood is dark red because of the hemoglobin, and its heart is also dark red because it has myoglobin. And so icefish don't express hemoglobin, making their blood look white or clear, and this fish in particular does have myoglobin, so its heart remains pink. but other ice fish also don't express myoglobin. So as you can see, its heart is this beige color, the lack of myoglobin, and its blood is clear. And icefish are the only vertebrates on the planet that don't express hemoglobin or myoglobin. So what is also super interesting is that all vertebrates also have these recently discovered globin genes called globin X, neuroglobin, cytoglobin 1, and cytoglobin 2. These all have an affinity for oxygen because they have a heme group and they're globin proteins. However, very little is known about the specific function of these globins, specifically in Antarctic fish. But it's predicted that they function to transfer oxygen to cells as well as to protect against oxidative stress. And these predictions are based on experiments done on other vertebrates. So the purpose of this study is to investigate the evolutionary adaptations that have allowed fish to survive in the frigid Antarctic waters, as well as to investigate how ice fishes have been able to survive without hemoglobin and myoglobin. And we hypothesize that in Antarctic notithenoids, the other globins evolved functions to further protect against oxidative stress. And we also hypothesize that in ice fish specifically, these globins evolved functions in oxygen transport since they don't have hemoglobin or myoglobin. So in order to study gene adaptation, we can look at how the genes and protein sequences have evolved within notithenoids. So I first retrieved the full-length sequences of neuroglobin, globin X, both cytoglobin 1 and 2, and myoglobin from 38 notithenoid species, including icefish, and 5 outgroup species. And I aligned all the sequences. This is an example of a partial alignment of cytoglobin 1. And from these alignments, I reconstructed protein phylogenetic trees using the program RAXML. And I tested for change in rates of evolution using the DataMonkey web server and the provided tests ABSREL, RELAX, and MEME. So for the sake of time, I am only going to be showing the gene evolution results of globin X. So this is the protein tree that I reconstructed with RAXML. And the test ABSREL did in fact detect an accelerated rate of evolution before cold adaptation. So as you can see, the cold adaptation branch is in blue, and where there was the most evolutionary changes is in this green branch right before we get to the Antarctic fish. And then the RELAX test showed that there was an intensification of evolution within Antarctic fish, so there wasn't a lot of changes that happened in the globin X gene between Antarctic fish compared to the temperate fish. And finally, the MEME analysis did detect 4 amino acid sites under episodic diversifying selection, which means that there were 4 sites that significantly changed throughout the temperate and Antarctic fish within the globin X gene or protein sequence. So I analyzed gene evolution adaptation using the gene expression analysis. And I first prepared cDNAs from the brain, heart, liver, spleen, kidney, and gills from 4 notithenoids. And these 4 notithenoids were strategically chosen because we wanted to compare temperate fish to Antarctic fish. So one is Codopercogobio. The next is Aleginops maculavinus. The third is Notithenia cordyceps, and the final one is Chiencephalus aceratus, which is an ice fish. So then I ran a reverse transcriptase PCR using gene-specific primers for neuroglobin, cytoglobin 1, and myoglobin. Then I ran a gel and imaged it and quantified the expression using the Biorad Image Lab program. And then I recorded the adjusted volume intensity of each band compared to the adjusted volume intensity of beta-actin. So the results showed in cytoglobin 1 that there was a lot of expression throughout all tissues of these 4 species of fish. And an example of a gel I ran is this is cytoglobin 1 in Sciaceratus. And so this is the ladder we compared these bands to. And this is where I recorded the adjusted flame intensity. And so what's really cool about cytoglobin 1 is that specifically in the gills, we see that it's only expressed in the Antarctic fish. And this leads us to believe that this is a cold adaptation because it's not expressed in the gills of the temperate fish. And then next we look at neuroglobin expression, and we see that there's mainly expression in the brain of all 4 species. But what's also really striking is that the expression is a lot stronger in both of the Antarctic fish compared to that of the temperate fish. And this also leads us to believe that this could possibly be a cold adaptation, and as well as preventing oxidative stress within the brain, since the brain is such a vital organ. And next we see myoglobin expression in the heart of Hemaclovinus and N. cordyceps, and not in Sciaceratus, which is to be expected because it is in fact an ice fish, but we don't see any myoglobin expression in the heart of C. gobio, which was really peculiar because it's a temperate fish and it should be expressing myoglobin in the heart. But if we go and look back at neuroglobin expression, we see that Cotoperca gobio also expresses neuroglobin in the heart. And we're speculating that this could be a compensatory adaptation for the lack of myoglobin expression in its heart. So to conclude, both the gene analysis and the gene expression analysis suggest that some genes may have evolved with cold water adaptation. For example, in globin X, we saw the green branch that did show a lot of evolutionary changes before cold adaptation. And in the gene expression analysis, we saw some interesting expression in cytoglobin 1 and neuroglobin. And as for the specific functions of these globins in icefish, the results are less conclusive and further research needs to be done. And so in the future, we're going to determine the consequences of the residue site substitutions from the meme analysis and determine whether or not there was a conformational change or perhaps a change in oxygen affinity from these site substitutions. And then we're going to finish the study of cytoglobin 2 and globin X expression in the previous tissues and species analyzed. And then we're going to confirm the lack of myoglobin expression in the heart of C. gobio, and we're going to do so by including more species to analyze that are closely related to C. gobio. And finally, I would like to thank both Thomas and John for teaching me the past 2 years and for giving me the opportunity to work hands-on in this incredible study. Thank you so much for listening.
Thank you, Audrey. Wow, that's a lot of data. Does anyone have questions for Audrey or anything from the Q&A?
Like we just got a question in the, oh, nope, we just got a thank you in the Q&A. So no questions in the Q&A right now.
I have one. Audrey, you did a lot of different analyses can you talk a little bit about your data acquisition and your analyses and how you decided on what analyses to use in the context of this data? Was it something that there was already previous work done with certain analyses being used, or did you come up or your team come up with the particular analyses and comparisons that you did with the other fish? Yeah,
so for the gene evolution analysis, I retrieved the sequences from different sites like Ensemble and Sequence Server, and I compiled them all together. And after I aligned them, DataMonkey provides a lot of parameters for determining if there are evolutionary changes between sites, and a lot of tests didn't show any significant changes, but there were there's a lot of different tests that test for different like rates of evolution, and so Toma helped me determine like what tests to do specifically for the gene evolution analysis.
And was this a project that you had started pre-pandemic, or did the pandemic affect any of your kind of work on this study?
Yeah, so I started working in the pasta weight lab fall of 2019. And so I began retrieving all the sequences then. And I also started the gene expression analysis not too long after that. But once the pandemic hit, everything was remote. And so I was still able to run these analysis remotely on my own computer, which was very convenient. So yeah. It's a long study. It's ongoing.
So you had support from the lab, but you were able to do it on your own as well.
Yeah.
Great. Thank you, Audrey. Are there any other questions from the panelists or in the Q&A?
Nothing in the Q&A.
Okay. Thank you, Audrey. And we will move to our next presenter, who is Lukas MacMillen, and he's in biology. And he's going to be talking about the condition-dependent recombination in flies, Drosophila, assays and implications. Lukas, it's all yours.
Thank you so much.
And Lukas, I think all of you are coming in right on time, but I will give you a hand up if at the 13 minutes, okay?
Sounds wonderful. All right. Hello, everybody. My name is Lukas, and I'm an undergraduate researcher in the Sing Lab, and we're in the Department of Biology and Ecology of Institute of Ecology and Evolution. And as Barbara just mentioned, I'll be talking to you about meiotic recombination and the theory of condition-dependent recombination in fruit flies and whatnot. And this is part of an ongoing literature review that we will be doing. And I've broken my talk sort of into 3 different sections. The first of which is going to give us some background on phenotypic plasticity, and it's going to highlight the prevalence of this in meiotic recombination. And then I'm going to go in and discuss the complexity of plastic recombination, and we're going to end with the condition-dependent recombination hypothesis and more on like the literature review and future implications. So as I said, going into meiotic recombination rates being phenotypically plastic and changing with the environment, we have to start with phenotypic plasticity, which is this idea that one genotype can produce multiple phenotypic responses depending on environmental conditions. So there's this variability within phenotypes from a single genotype. And this is shown first in a dung beetle as an example, where there's this horn size variation depending on their larval nutrition. Also in water fleas, there's this helmeted phenotype, which is shown on the left, and this can be induced from predator presence. And a similar predator-induced change is seen in these anurin and tadpoles, where they become bulkier when predators are within the region. And most importantly for our discussion today, we also have meiotic recombination being shown as phenotypically plastic towards just environmental cues. And you may remember meiotic recombination as this crossover-mediated sort of genetic exchange. So here's the crossover and then some genetic exchange that occurs during sex cell production, or as I have it up there, like immunogenesis. And for more than a century ago, just in general, Plough and colleagues identified that there was this crossover variation across temperature gradients. So we have crossover frequency increasing across the y-axis and temperature increasing across the x. And what this really showed is that crossover frequency was phenotypically plastic, and thus meiotic recombination was also phenotypically plastic. And since then, since that was 100 years ago, there's been a plethora of environmental cues that have also been associated with this phenotypic plasticity in meiotic recombination. And I guess a slight note is that all of these papers that are highlighted here are in Drosophila melanogaster, but there's plenty of other literature and other supporting model organisms. So now coming back, we kind of know that matter recombination can respond to environmental cues in this phenotypically plastic manner. And we kind of have to ask, why would it evolve in the first place, which is still an unknown question. And we're going to go through this little diagram that I've made to understand the complexity of plastic recombination in general and why the evolutionary history is so important to uncover. So here I have what I'd like to say as two alleles. So you have to use your imagination, one with Apple Messenger and an operating system, and the other with the Samsung Messenger and its operating system. And we can also say or imagine that you're a really big fan of the Apple Messenger app, but you dislike its associated operating system, and then vice versa for Samsung's side of the equation. One positive side of recombination is that there's this ability to bring together these favorable allelic combinations. So you can imagine a crossover taking place between the messengers and operating systems for both of these, thus isolating the favorable and the unfavorable components. But then you have to question at the same time is, what if you are an all Apple everything person, or from the biological sense, what if the arrangement of your genetic information was already ideal? And that's the key point here, is that just as easily as a recombination can bring together these favorable allelic combinations, if you already had your favorable combinations from the first place, it can take them apart. And that's sort of that real complexity, is that with plastic recombination, you can have something that responds to the environment, but produce both positive and negative effects. So how would it evolve to kind of mediate that? And we're now going to go into the condition-dependent recombination hypothesis and show why it is a promising hypothesis on the evolution of plastic recombination and why our lab has been interested in it. And starting with it, the condition-dependent recombination hypothesis is centered on this idea that plastic recombination varies with organismal fitness. This is also known as the fitness-associated recombination, or FAR hypothesis / model. And I want to note that fitness here is biological fitness, which is just this ability of an organism to survive to a reproductive age, find some mate, and then reproduce offspring. And I have this simple graphic to just kind of highlight the main assumption of the FAR model, and that is that organisms at low fitness will be having, I guess it's important to say that fitness here is increasing across the x-axis, and recombination rate is assumed to be increasing across the y. So at this lower fitness end, we're saying that recombination rates are increased because those chance benefits of bringing together those favorable allelic combinations outweigh the chance cost of breaking apart any favorable ones. Whereas in a high fitness individual, we have an expectation that recombination rates are occurring less frequently, or just decreased, because there's now an increased chance of losing or breaking apart these favorable allelic combinations. And with this model, and this sort of simple assumption made by the model, our lab has focused on two main questions. The first of which being, what evidence supports the FAR model's expectations, so the graph on the left, and the second being, how have empirical studies measured the effects of fitness on recombination thus far? And I'm not going to go into some of the key points that were selected from the review, at least for Drosophila and melanogaster. And we're going to run through most of the findings, but I will focus on one single finding afterwards, just because it's more palatable and digestible that way. So just generally, this is the one that we'll go into more in depth soon. But the first point was that FAR support was found from organismal fitness, where organismal fitness and recombination rate were shown to vary across wild type genetic background. And another study found that deleterious or harmful mutations affect fitness and recombination in a manner that was concordant with the FAR model's expectations. And then lastly, there was this increased heat shock exposure time, which was shown to decrease organismal fitness and increase recombination rate in a FAR concordant manner. So now, as I mentioned, I want to go into depth on this 1981 paper, and the 1986 is fairly similar, just with a few other assays used. And this one was chosen because it has a fairly palatable graphic. So what these researchers did is they identified 70 unique wild type genetic backgrounds in nature, and they isolated them. And they did this because they wanted to know whether or not genetic background would affect recombination rate and organismal fitness. And then what this graph is showing is the summary of the results. So here we have fitness increasing across the x-axis, And we have the recombination rate increasing across the Y. And each of the names on the right side are just assays used for data collection purposes. And we'll go into each of them specifically, but I wanted to note that these little shapes in the graph, so the circles and triangles, are what are called fitness classes. And this was basically saying that of these 70 genetic backgrounds viewed, each is placed into some corresponding fitness class, with the fitness class being defined per assay. And this is just for purposes of data analysis. So going through the study's results, we first have viability data, which is collected by comparing the proportion of progeny produced to that of an expected value given Mendelian genetics. And they're using it here as a fitness measure. And what they show is that the trend line represents that viability assays of fitness produce a correlation that is non-concordant with farm model expectations. So there is not this as fitness is decreasing or increasing, recombination rate is decreasing. Whereas with male fertility assays, so measuring the success rate of male copulants or sex, as they determined this as females that had sperm left over after an opportunity for sexual encounters. The researchers found that there was a significant correlation in agreement with the farm model's expectations, though one thing that's kind of interesting to note about this view of male sex rate is that it's not really measuring the percentage of fertility for a male sperm. And so that's just interesting in terms of biological fitness. And the other thing that's pretty useful to know is that there's only one asterisk here because it shows this farm model expectation, but only for certain regions of the second chromosome, which is what they were examining under. And then the last assay that they used, which was female fecundity, and this counts the total number of eggs laid per female. And they found another significant correlation that's in agreement with the farm model. And this time it was over a much larger range of the chromosome that they were studying, so the second chromosome. But bringing all three of these assays together and also inserting the very simplified expectation of the farm model, we know that data from 2 of the 3 assays yielded this negative correlation expected by the farm model. And the main takeaway from this figure is this idea that not only did they find that genetic background affects both organismal fitness and recombination rate in the far expected manner, but it depends on the assays of fitness used to gather data, and it portray this relationship. And these sorts of trends highlight the promise of the farm model, and it also shows kind of what assays are significant, or maybe questions what assays might we use in the future to further test the farm model. And so that's where we kind of have to ask what's next in the field of research and trying to uncover the evolution of plastic recombination. And to kind of go into this, I want to present to you all the papers that we predicted sort of at first glance, first pass, to have some discussion of the farm model and condition-dependent recombination. And then these papers in red are what I like to call the not so condition-dependent recombination papers, because though they're very accurate and fair pieces of primary literature, they didn't directly assay the farm model for some reasons. And for some of them, it was this idea that there's this lacking standardization in fitness assays, which just made it so that there was no availability for measuring the FAR analysis, or they just overlooked it. And in some other cases, there's also an assumption that environmental cues were acting stressfully on an organism, which also causes this lack of FAR analysis, because in order to test fitness, we can't just assume that something is being inhibited or being decreased or increased by some environmental cue. We have to have some sort of visible effect of it. And just a general conclusion is that both fitness assays and standardization and this redefinition of stress, which one way could be through the introduction of these novel bioindicators of stress, are necessary next steps as we look to elucidate or uncover the evolution of plastic recombination more broadly, and then more specifically, the validity of this FAR model and testing how accurate it is at describing the evolution of plastic recombination. And then I have a few acknowledgements. So I have the Sing Lab, my mentor, and everybody else in the lab. I also have the McNair Scholars Program at the University of Oregon, and then Oregon undergraduate research in SPUR for some summer funding that they also facilitated me with. And I guess I will open it to questions and stop share.
Thank you, Lukas. That is also a tremendous amount of data. So I'm going to open it for questions. Anything from our other participants or from the Q&A, Marie?
We do have a question in the Q&A. And the question is, just out of curiosity, have you as a lab discussed any possible mechanistic explanations for how recombination rates could be altered by fitness?
In terms of the mechanisms, I haven't, off the top of my head, mechanisms for how fitness would then interact with the genome to induce recombination is not coming off the top of my head. But I'm sure from my experience in like genetics classes, there are a few ways that I would approach this, but they are also blanking my mind at the moment.
I have a question for you, Lukas. So this involved looking at a substantial literature review. And this is a very commonly done set of assays, and fitness and is a common topic in the area of fruit flies. So can you reflect on how doing that literature research and that literature review, and thinking about the fact that you were questioning the how these reviews were done? You were just not taking them at face value. You were actually analyzing whether or not they were clear about their assays. And this is what I understand, that you're asking whether they're clear about their assays, whether they really did test using the fitness tests that were and that some of them are not standardized. How do you reflect on that in terms of how you think about science maybe differently than you did before you did that kind of complex analysis and critique of these various literature reviews?
So for quick clarity, the literature review is sort of the process that our lab is doing as a group. So we're writing the literature review based off of individual pieces of primary literature. So
Right.
To clarify your question, it would be—
So the primary literature is what I'm talking about. So
okay.
After you've done the literature review of all these primary pieces, yes, thanks for that clarification. How do you think, are you thinking differently about how science is done and how it should be done?
I think that I had anticipated a wet lab experience, but switching to just analyzing primary literature has been very valuable in terms of building my, like, how I would approach any wet lab experiences and asking questions and understanding, especially like, what does it mean in terms of how are my assays being set up and all of that. So I think it's maybe a lot more critical, yes, at thinking like beforehand about what I'm doing and looking at the implications of that. And I think that a lot of, because you're also asking my interpretation of just how publications are published and that stuff. So like the critiques of literature in general. I think that I've gotten to experience some of the critiquing process in the lab, which has been nice. And almost all of these pieces of literature were useful and very accurate in terms of what they were asking. And it's more discerning whether or not there's enough to say on, like, or to input to give in on the FAR model, like specifically or explicitly. So it is very interesting to be extremely critical about literature when maybe they're not even asking the questions that you are asking. So I think that's been really fun as well.
And I assume this was partly because of the pandemic, that you were no longer able to be in the lab. So you're kind of doing, instead of being lab focused, you were primary document focused. And that really kind of changed you as a scientist a little bit. That's interesting.
Yeah, I like to say it was on the fly because we're a lab, and it's really funny to do so. But yeah, my PI, Nadia, was very quick, very on the fly at making this idea of a literature review that the lab could partake in, because yeah, COVID lab access has kind of put a hamper on accessibility and like just the wet lab experience in general.
Okay, thank you. It's really interesting to have you all reflect on how the pandemic has kind of altered or not altered your research path. Are there any other questions for Lukas? Anything, Marie?
No other questions in the chat.
Okay. Okay, thank you, Lukas. And we are moving to our final presentation of today, and that's Isabella Garcia in biology, and she is a visiting McNair Scholar from Sul Ross State University. And Sul Ross is Alpine, Texas, correct, Isabella?
Yes, ma'am.
And I've actually been to Sul Ross University as I voyaged through West Texas. So really happy to have you. And Isabella is going to be talking about the reproductive investment of native versus invasive cane toads. So it's all yours, Isabella.
Hello, yes. So I did my research specifically on the species Rhinella marina. Previously, before this, there was a switch between Bufo marinus and the species that I looked at. I did this research through the McNair program at my university with my mentor, Dr. Crystal Kelleher Graham. Okay. So to start off, this is important to address just what an invasive species is. And these are just organisms that are introduced outside of their native range. And in most cases, they have pretty negative impacts on the economy as well as the environment in which the areas that they're introduced in. And during my literature review, we kind of categorized between about 4 different areas that they have impacts on, including competition, hybridization, predation, and the spread of different pathogens and diseases. In terms of competition, it's been shown that invasive species just outcompete native species for resources, primarily within retreats, food, and space. In terms of hybridization, invasive species interbreed with native species, and this ultimately leads to a reduction in the gene pool. And this has been specifically shown from an example would be like the green iguana, which has hybridized with the blue iguana that's native to the Cayman Islands. This has led to even further endangerment within the species. And this is just an example of how invasive species impact their introduced ranges. In terms of predation, invasive species tend to just out-eat and out-compete for a variety of different resources in comparison to native organisms. And good examples of these different types of invasive species could be like feral cats or feral foxes. And so one of the main things that we looked at and that really influenced this research is known as the enemy release hypothesis. And this is really just an idea that invasive species can escape their native range enemies. So these enemies could be competitors, predators, as well as just different types of pathogens and parasites. And the loss of enemies in the invasive range leads to lower parasitic densities and the ability to invest more in different investments. So this could be like reproduction or dispersal traits. So now we're going to look at the specific species that we looked at in this research, which was Rhinella marina. And this is formerly known as Bufo marinus, and they have been known to establish in a variety of different areas, and they've become invasive in a lot of different places. They're native to South America, and the species that we looked at was Guyana and French Guiana. They've also been introduced across the Caribbean, including the Pacific Islands, all the way up to Australia. And some of the main factors that I came across doing my literature review is that they've been able to successfully establish mainly because of their absence of native range predators and competitors, as well as selection and dispersal traits. And they've also been shown to just be able to show risky behavioral traits. And specifically within dispersal traits, cane toads that are invasive have been shown to have greater tibial lengths as well as streamlined body shapes, which help with speed. And that allows them to ultimately disperse at a lot faster of a rate. And in terms of risky traits, there's also been research that has shown that they take directionality of dispersal, they show more boldness, as well as more risk-taking behaviors. Okay. So for this research, I wanted to compare invasive versus native populations. Considering the life history of cane toads, I proposed that populations would invest more in reproduction than native populations. And this would allow them to effectively spread and colonize at a faster pace. And more specifically, I hypothesized that cane toads that were invasive would invest in larger quantities of small eggs. And this is primarily because smaller eggs would be able to fit in a given space, which would allow them to be more successful within reproduction. So now we're going to get into some of the methods and materials. This part of my research was greatly impacted by COVID-19. So we can move on to the next one. Thank you. So to start off, these cane toads were analyzed from 3 different invasive populations, and they were compared to 2 native populations. We looked for invasive populations, we looked at Australia, Bermuda, and Hawaii, and then we compared them to native populations, Guyana and French Guiana. Luckily, these were collected before COVID-19, but they were collected in the wild. So they were euthanized. My professor used clove oil, and then these toads were dissected, where we were able to remove the ovaries, and they were rounded to the nearest 3 decimal places. And then once this research was done, I had the ability to measure the mean subsample, which was about 0.635 grams per ovary. Okay. And so the main materials that I used in this, which worked out once COVID hit, was I was able to just have these pictures ready to go. And I used the cell counter plugin in ImageJ. You can kind of see that picture in photo 3. All those little yellow dots is literally, I manually counted each egg in this photo. And I was able to do that with the Wacom bamboo and pen and mouse, which is in the photograph 4. And then we used ANOVA tests. The log transformations variables that were used to ensure normality of the data included the total number of eggs and the mass of each female toad. So now we're going to get into the significant results that we found after months of manually counting thousands of eggs. Okay. So one of the main results we found was that cane toads in their invasive ranges had more eggs than within their native ranges. And this ultimately concurred with the hypothesis. Unfortunately, with what I had hypothesized, that smaller eggs would fit in a given space and that invasive cane toads would invest more in smaller eggs. But there was really no differences in the mean egg size between native and invasive populations, which was pretty interesting and did not concur with what I had initially anticipated. So we also had some positive correlations in terms of body mass and the number of eggs. We found indications that heavier toads had more eggs than smaller toads. And this isn't really a comparison between native and invasive. It was just something extra we went ahead and looked at. And we also found positive correlations between mass and egg size. And this was ultimately showing that heavier toads had larger eggs. Okay. So in terms of these results, what we found was significantly higher numbers of eggs in their invasive versus native populations. And this concurred with the hypothesis that invasive cane toads would exhibit larger clutch sizes. Now, in terms of egg size, again, I had initially anticipated that invasive species would invest in smaller eggs to support the increased number of eggs that they would be producing. Unfortunately, that was not what happened when we actually did the analysis. This ultimately suggests that it may be a less focus on egg size compared to egg number in exchange for an increased fitness in stressful conditions. A possibility that this may be showing is bet hedging, which is a split resource. So there may have been the possibility that invasive species were instead of just producing eggs of all the same size, they may have potentially been creating different sized eggs, so some smaller than others, and that's a possibility. But unfortunately, we weren't able to look at that because I did not take the pictures on the same scale. So when I was taking pictures of the eggs, we had laid them out, and not on all of the pictures, depending on the population, I wasn't able to take them on the same scale because of, camera issues and things like that. So one of the main shortcomings for this research was mainly permitting and ethical issues, specifically in Guyana. So their cane toads are native in this specific area, and we had a limit on how many we could catch in their native range. So the number was very not proportional to the sample sizes of other populations that we included in this study. So for future research, I think we could focus on greater sampling sizes in native ranges to kind of get a better number ratio. And in terms of further investigations, I think there could be different topics on potential trade-offs between reproduction and other energetic investments, which could be anything from like dispersal traits and things like that. So this is the last slide, and this is really, I just want to acknowledge the McNair Scholars Program for the opportunity to even conduct research, as well as my mentor, Dr. Crystal Graham. It was a very difficult research project to conduct with COVID, and I'm very grateful that I even got to finish. And I'd lastly like to thank my mom. So, all right, the last few slides are just the citations, so we don't have to show that.
Well, thank you, Isabella. Boy, that was really an incredible field study combo with intense data analysis. And you made a comment that it was affected by, when you started showing us the eggs, that it was really affected by COVID.
Yes.
Can you tell us a little bit more about that? So you had taken the photographs of eggs, but do not plan to necessarily count them in the same way that you ended up having to, or what was the difference?
So for some of the actual capturing of the eggs, I was not able to do. So my mentor traveled to the last two places. Luckily, she's from Australia. So most of our numbers were came from that. It was very rushed during this time. So I honestly, the idea of taking them on the same scale completely, I didn't even think about it. And it was very rushed, and yeah, so
part of it was that different people were taking the photographs.
Yes.
So you had that experience of what do you do when you're working on an international team and different people choose to take them at in a different scale.
Yes.
So I think that's a really interesting international research, or when you're doing research with remote, you have to communicate so much to each other, right? Thank you.
Yes, ma'am.
So I will open it for questions for Isabella. Anything, Marie?
Nothing in the chat thus far.
I have a second question, if there's nobody that is ready to speak. Isabella, can you talk a little bit about this idea of riskier behavioral traits? It's interesting that is what makes these people, the toads as people, that makes these animals more successful in their invasive environments, in the environments where they become invasive. But it's interesting that those riskier behavioral traits seem to also be potentially dangerous or not as positive for the species themselves. I thought it was interesting that you talked about the kind of different sizes of eggs that they might produce, for example. Do you get any sense of if these riskier behavioral traits actually potentially negatively affect the invasive species as well as potentially are positive?
So that specific research was mainly in Australia. My mentor actually was the one that conducted that research in Australia, where they kind of tracked the cane toads and kind of looked at the areas that they were dispersing. But really what they found was that they did take riskier traits, like they were expanding into areas that they weren't really suggested, like it wasn't anticipated that they would be going into a lot of the drier areas and things like that. But a lot of those factors included, like, for the most part, most of the other predators in the area could not actually eat them. So cane toads are poisonous. And so like the main, like, for example, I cannot remember off the top of my head the type of snake, but it's known to eat a lot of the native frogs and things like that. But it was the cane toads were actually killing the snakes when they would eat them. The poison would start to kill the snakes. And so I think that a lot of the directionality of dispersal was really just that they had no competitors, and that's something that they found, was that like there was actually in specific areas where these snakes were actually starting to decrease in their population size because a lot of them were dying trying to eat these toads. It was really interesting, and there's a lot of different factors, and really it's really interesting. I think king toads can be a beast. So
yeah, I can see that. It's thinking about the different traits that make them successful, especially in different environments. It's really interesting. Yeah. Any further questions for Isabella? Well, thank you, Isabella. We really appreciate you coming to join us. And I'm going to open it up for a full discussion here for a few minutes with all of your presentations. We have, it's just about a few minutes before 3, and we have to close at 3:15. But I'd like to engage in a little bit more open conversation. If we can all just be participants, and if we have any comments from the Q&A, Marie, would you let us know?
Absolutely.
Okay. So let's see. I'm going to put everybody so I can see everybody well. Thank you. Well, thank you, everybody. That was wonderful. It's really exciting to see the research that you've been doing and the focus that you've been able to continue with throughout this year. And also your reflections on how the pandemic kind of affected what you were pursuing, given all the variables that can happen with research. Small things suddenly become major issues when you can't travel, or you have a lab that's closed or something like that. So it has been a challenge, and I give you a lot of kudos for, like you say, getting through it and actually being able to finish the research. You are welcome to ask questions of each other, and I have a couple questions that I'd like to just have you reflect on and share your experience. So I'll open the floor for anybody who wants to ask one of your colleagues a question before I open it wider. Okay. Okay, so seeing none, I just wanted to ask you all to just kind of reflect on, now that you've done this research and you've done the initial work, you've done the analysis, you've thought about the conclusions that you can come or where it needs further research. What would you have done differently that you can think of that would have been useful if you would just reflect on it? What thing or things could have helped out or could have changed the way you approached your research? And I know a piece of that is going to be the pandemic, but sometimes there are other pieces as well. So could you each reflect on that? I'm going to maybe just, I'll go around in terms of my my screen. Lukas, can you reflect on that?
Sure. I think so I was like guaranteed to be doing something over the summer. And I think it was like, I can't remember exactly, but maybe like 3 weeks when we knew exactly that we would not have any in-lab option. And so then we really went to like the literature review. And so I'm not sure if there's a ton that I would do differently during the literature review. I think maybe I would have spaced out my readings a bit more if I did it like that, just because there's like so many papers that we had to digest. But it was pretty good for the circumstances in terms of like what we could actually do. And I mean, if I had a little bit more time to create a completely new project, I probably would have done something more like computationally. So coding and making stuff like that would have been really fun. But yeah.
Okay. Audrey, what about you?
Yes. Well, having hindsight because of the pandemic, I probably would have prepared my materials better to take everything remotely, because not having like all of my data or all the files I needed to do all of the evolutionary analysis was difficult because it made the process a lot longer. But that's just because of the pandemic. I think for the actual expression analysis, for myself, being more in tune to the details and like the nuances of the different levels of expression from the get-go would have been helpful for me to piece together everything that was happening. Yeah, having a little different mindset, I guess.
Thanks. Isabella, what about you? Anything that struck you?
Yeah, I would have to agree with Audrey about having been more prepared for getting your materials together. It was really difficult trying to, my professor had to mail me most of my stuff. I'm originally about like 6 hours from my university. So that was difficult, having to get mailed materials and things like that. But really it was just also the difficulty of communication to try to make sure that everything was being done properly and the right way. It was kind of hard also to like get to different places during the wettest time of the year, since cane toads are more reproductively active during the wet seasons. I wish that I would have been able to go to all the locations and be able to collect during the wettest times of the year. And that's different depending on whether it was in Africa or Australia, so it was difficult, and it really prolonged the process for trying to collect everything. And I wish I would have had more time to be able to actually go to these places. I was really excited. So yeah.
Yeah, the adventure wasn't quite there.
All right.
What about you, Emily? Anything that you would reflect on?
Yeah, sure. Looking back, I think I would have started learning about our studio a lot sooner and learning how to create figures really well, and how to manipulate the data that I had. And I also think it would have been really helpful to have maybe like another undergrad or somebody who could have helped me sift through a lot of the data. It was really similar to Isabella's project, where I was in ImageJ calculating surface area, one blade of grass at a time. And it's just like totally wild how many hours you can spend doing that with just one person. So unlimited resources. If I could have done it differently, I would have definitely, had another partner to help me with some of that initial data uploading.
I think sometimes, given what we've learned during the pandemic, is just having a partner, a team, to keep you going, to keep you encouraged. That's part of what happens in a lab or a team scientific environment, is you have encouragement, and it was much harder to come by, for sure. I just have one other question that I always like to ask students, because it's one of the things that I really wish somebody had talked to me about when I was thinking about what I wanted to do. I was a bio major and had a specialty in biopsych, and then became a medical librarian because I was interested in the informatics side of it, which really turned out to be the right thing for me. And one of the things that happened to me in making that decision was I did an experiment with rats in the basement of the science building. And I was looking at pheromone production. And I got bitten by this rat. But I found out, and it wasn't just being bitten, but I found out that that kind of research just really wasn't what I was interested in. And I was allergic to all things plants, so I knew that I wasn't going to be in plant biology and field biology. What I realized was the thing that fascinated me most about science was the literature, the thinking about what the primary sources were saying, seeing how the literature reviews were done. That's what I was fascinated about, the kind of publication of information. And so I went the informatics way. But it was a struggle to figure out that that was really what I was wanting to do. So I was just curious if any of you would be willing to talk about what you found you really liked about the research you're doing, or disliked, and not just the COVID side of it, but the intellectual lab or research analysis that you do, and how you're thinking about it in terms of how does that fit into your future. Would any of you be willing to talk about that? Should I
share?
in?
Oh, I can go real quick.
I'll start.
Sure. I think that primary literature is a really good way, like when you have to read a lot of it, especially to figure out if you want to be married to that subject, kind of like you were mentioning. And I think it's definitely like probably a faster way to figure that out than doing like a wet lab project, because those can take a lot more time, and then you get just like one result that tells you whether or not you're interested, which isn't enough. And so I think I valued that a lot. I think that it has shown me that I like the application of interactions a little bit more. So, I mean, my research trajectory is more towards bacterial host interactions. So taking some of like the evolutionary genetics from this lab, but applying it more in terms of like, how do species interact with each other and like any type of host. So I think that's where it's brought me. It's definitely like helped me quickly figure out what is more ideal for my, long-term research goal.
Okay, thank you. Audrey, are you willing to reflect on that?
Yeah, definitely. For me, I think what interested me the most about this research was, well, you have like a gap of knowledge of this big mystery, like why something like this, and it's especially with like hemoglobin and stuff. I'm a human phys major, so stuff like that really interests me, and how like not only like human bodies work, but other animals' bodies work. And it was really fun to like kind of begin to fill that gap of knowledge by slowly like piecing together what possibly have happened along the way, like evolutionarily. I think the part of the research I enjoyed the most was the wet lab experience. I enjoyed going in as much as I could during the pandemic, and like collecting data and just like seeing the results, it was really satisfying to see that.
Okay, thank you. Isabella, what about you? What did you really like and about the project, and what are you thinking about future-wise?
I really just liked that I feel like I learned so much. Nobody in my family has ever done anything like this before, and I had a great mentor that kind of also took me under her wing for an independent research study after I had done this one. So this one was looking at reproductive investment, and it got me really interested in like the immunity aspect. So I did another short research regarding the actual comparison between invasive and native immune systems. So I like really did a lot of white blood cell counts, so more counting, but that it made me really interested in like the immune system and how it works. And, you know, I know that I wasn't looking at humans and how we adapt to things, but I think it's kind of geared me towards more like parasitology type research in the long run, doing that. So along with this, because, you know, looking at the difference between presence of pathogens and parasites was really tied to the enemy release hypothesis. So I'm really interested in that and how it actually influences people in the long run. I think that's something I'd be interested in. Yeah, so
Great.
I've had some interesting talk to talk about.
So yeah, from toads to people, right? Exactly. Thank you.
Yeah.
Emily, what about you?
Yeah, I think doing this research has been super, super interesting in so many ways. I think previously I have been learning about plants more as individuals and like thinking less about ecology and relationships, you know, within species and then between species. And so it's been super cool to learn about, you know, this very specific grass species and then think about like the minute details that go into that and then zoom out and get like this really broad view of thinking about what's going on in a whole region of like the Great Basin and thinking about like how different factors like climate change and fire cycles are going to be changing in the future. And so I think just as a whole, it's given me like a really even more interest in restoration ecology and not so much just like one specific plant that I might like be really interested in, but more of just like a system and how it works as a whole.
Great. Well, it's really wonderful to hear your research and your reflections. I feel like, you know, even though we've been through a very tough year and a half, it's kind of useful to think about what we have kind of learned about ourselves and our research along the way, because it's kind of been forced upon us to think kind of differently or do things kind of differently. And I really want to also reflect on your great mentors and your faculty mentors that you've been working with. It sounds like they've really stepped up in many cases as well to give you a positive experience during these difficult times. So I thank you so much for sharing with us today. And we're close to our time limit, but I think you all know that the recordings, this session will be up on YouTube. It will not be immediate, but it will be up there in the next, they're getting them up in the next week or so. And they look forward to you sharing it with your friends and family to see what you and other students have been working on. So Marie, do you have any final words?
Just that I'm so impressed by all of you and congratulations on your research and your wonderful presentations today.
Yes, and I second that. Congratulations and thank you for sharing with us. Take care.
Thank you.
Thank you.